All right, great. I'll begin. I do apologize, everyone. I'm unable to hear anyone. So if you do have any questions, please tap them into the chat or ask one of our representatives from NanoString in Korea or our partners from Philly, Korea, and they can ask me via text. So I'm not sure if they did an introduction, but my name is Marshall Fettrell. I'm the Senior Technical Specialist for the APJ region, and I'm based in Brisbane, Australia. And it's really great to have a conversation with our Korean customers. I really love Korea. It's an amazing country, and you certainly understand spatial biology, and the research outputs that are coming from Korea are simply amazing. So it's very exciting to talk to you, and I'll be in the country next month, and hopefully I can see some of you in person, which would be great. So just let you take that off there. There we go. So just to give you an overview of who NanoString is. So we're a biotech company that was launched out of Seattle, Washington in the US. And we're a research-driven company. So we build and we develop our own chemistries and our own systems. And we've been doing that since 2008 when we launched our Encounter analysis system. So, there'll be, I think there's over 1,000 publications actually out of Korea alone, just on the Encounter. So this is our bulk gene expression analysis instrument. And we designed it with very simple chemistry. So we can do, there's no enzymology, no amplification, and we can do direct molecule counting. And we can do that in FFPE tissue. And it's really the foundational chemistry that we used for the development of our spatial biology platforms. So if we fast forward to 2019, that's when we launched the Geomix digital spatial profiler, really, revolutionized the way that we can interrogate tissue. And then, of course, just Q4 of last year, we launched the CosMx Spatial Molecular Imager, which is our single cell and subcellular system, which I'll be spending the majority of my talk on today. But also our atomic spatial informatics platform. So this is actually not just a software, but an interactive portal that you can use to interact with your Geomix data and also with your CosMx data, and I'll be talking about that. So globally, we have over 1,500 instruments installed, nearly 7,000 publications, and over 130 support experts to help you with your research. And in Korea, of course, we also have our distribution partner, Philly, Korea, to help you in that endeavor as well. So CosMx, this is our new single cell again platform, is winning attention and accolades in the academic space, of course. So we reached the cover of Nature Biotechnology later last year, and we were one of the top 10 inventions of 2022 in the Scientist magazine. Even outside academia, we're also gaining some recognition in Alzheimer's research. So this was a publication in National Geographic, of all places, in the latter half of last year as well. And this is coming from Dr. Miranda Orr's lab out of Wake Forest Medical School in the United States. And in this example here, you can see this beautiful image of a human brain section. And this is looking at a 6,000 plex RNA panel, 120 proteins. And what Dr. Orr was able to do was identify these senescent or zombie cells in the human brain. And these cells, they accumulate tau protein and they release toxic substances that can lead to an advancement of Alzheimer's disease. And she's using our platform to understand how to target these cells and develop novel treatments. So we're really excited to see some other customer applications of this panel when it rolls out later in the year. So our chemistries, this is both of them. They use a shared in situ chemistry. So this is what we've been working on from the very beginning of our company. And on the RNA side, we use a hybridization game. Right. So on the Geomix side, we have very, very short probe sequences, so just 35 to 50 nucleotides. And on the Geomix side, we're just targeting along the most conserved regions of an mRNA transcript. On the protein side, we're using a series of antibodies. But how we quantitate either of these analytes is by capturing these known oligo barcodes that are covalently bound to the ISH probe or the antibody via a UV photocleavable linker. And how we quantitate those is with a sequencing component. So downstream, you'll collect these barcodes and you'll create an NGS library, and that's where you do your quantification. So it's a digital output using the Geomix instrument. And this can go up to whole transcriptome. So we're capturing these probes directly in situ, directly out of the tissue, and we can go up to really high plex. So 22,000 plex is what we can reach, 150 plex for protein for Geomix. On the CosMx side, on the right hand here, so very similar in terms of the in situ hybridization probes that we use for that mRNA transcript. So very short sequences, again, 35 to 50 nucleotides. We tile about 5 of those along the mRNA transcript. But the readout side now, instead of using sequencing, we have a readout domain. It's roughly 100 nucleotides. And we have 4 of these individual landing pads that you can see on the readout domain. They're about 14 nucleotides or 14 base pairs. And what we do is we go through a series of in situ hybridization and imaging. So it's actually an image-based quantification of these particular analytes. And we go through 16 rounds of that. And in 4 of these rounds, we'll have an on event where we'll have a reporter and a dye that will fluoresce. And then at the end of that 16 round sequence, we'll have a 16-bit barcode that corresponds to that individual RNA. On the protein side of things, of course, we use a series of antibodies. We can get up to 64 plex. And we use a similar type of approach with the readout domain. If you're interested in doing more of a deep dive into our chemistry and application, these are our 2 foundational papers that were published in Nature Biotechnology, and you can access those via PubMed or just ping me, and I'm happy to share them with you. So both of these platforms, the way that we develop them is that they use industry standard tissue processing. So if you're familiar with an ISH type of protocol or IHC, multiplex IF type of protocol, that's exactly what you'll expect out of the Geomix instrument. So we don't have any proprietary slides. You just use a positively charged slide out of your histolab that you're accustomed to. Very large scanning areas for both. You fix your tissue to that, and then you'll run that on the CosMx or Geomix system. Both of the instruments were built to scale. So they can house up to 4 slides, and then we built them that way for a reason, and we want to use all of those 4 slides, currently and moving into the future, particularly with CosMx. And then we do our quantification. So again, I've already mentioned how we do that with both systems, and then both of those data sets can be integrated into a central place on a cloud-based solution with our AtoMx platform, which I'll talk about at the end of my talk. So as I mentioned, you need a positively charged histopathology slide, and you have a lot of flexibility in terms of the types of tissues that you can run, right? So you can use very large tissue sections. You can put multiple tissue sections. So here we have mouse brains. And for our Geomix instrument or instrument users and TAP users in Korea, the TMA or tissue microarray is the most important and most useful one for our customers out of Korea. So not only does this allow you to increase the throughput of the number of samples that you can profile on our system, but it also is a very cost-effective way to profile tissue spatially. And we can do this with both the Geomix and the CosMx. So what we found, over the past couple of years, based on demand and where the technology sits, is that the questions that researchers are asking, they interrogate tissues on different levels, right? And the type of platform that you use, we don't have a single technology that can do everything just yet. But it really depends on the question. And at NanoString, we have complementary solutions for that with the Geomix and the CosMx. So with the Geomix instrument, this is a multicellular approach. So we can look at populations of cells, and we look at them and we look at specific structures in the tissue that we can profile. So there's no single cell organ in the body. We know that they're comprised of heterogeneous cell populations. And what we can do with the Geomix instrument is outline those structures in the tissue, and we can collect very high plex. So this is whole transcriptome, 150 proteins. So you can do pathway analysis and understand what's going on. And then on the single cell side, or if you're interested in single cell type of applications, we have to use a spatial molecular imager, an imaging-based approach. Now, the issue that we have with that currently, although we're solving that problem, is the plex level that you can reach due to optical crowding and spectral overlap that you'll find with microscopes. But at NanoString, we have the highest plex on the market. So we have an on-market 1,000 plex product, and we've just demonstrated up to 6,000 plex, which is very exciting. So how you can see that applied in the tissue, here we have a sagittal section of a mouse brain, and we're highlighting the hippocampus here. So this is important for learning and memory. And for researchers who are interested in this particular structure, which you can see is very complex and folded, with the Geomix instrument, we can outline that specific tissue to a high degree of sensitivity with no gaps in between the tissue. So if, for instance, if you're using a microarray kind of sequencing approach, there's gaps in that tissue, and using a gridded array, and you might not capture all the information that you need. And if you're interested in the individual cells that comprise that particular structure, that's when you would move over to the CosMx SMI system, and then we can outline those particular cell populations and understand the interactions of each of those. So with the CosMx system, what we can do is look at single cells. We can do cell atlasing, cell interaction, receptor ligand kind of approaches. And on the Geomix system, it's a multicellular cell population approach. And that's what we'll focus on first. We'll focus on the Geomix instrument. So the Geomix can perform up to whole transcriptome, as I said, and it's a biologically driven approach with a high degree of flexibility. It's highly sensitive and cost effective because you select the areas on the tissue that matter to you most, and you're not throwing away your sequencing dollars on extraneous kind of portions of the tissue that don't matter. You can profile any sample on it, so FFPE, fresh frozen, whole mount, TMAs, organoids, and spheroids. As long as you can fix it to a tissue, we can profile it, even bone tissue. It's truly high-plex multiomic, and it's a scalable throughput depending on the design of your assay. You can profile up to 60 samples per week at any plex level. So what's unique about the Geomix instrument is its ability to use these digital micromirrors that are on the system. They're comprised of millions of tiny little mirrors, and what that allows us to do is outline these non-contiguous regions of tissue. So we have a colorectal cancer sample here in the middle, the tumor stained in yellow, and we have a CD45 stained in pink. And what the system can do is it can outline those specific tumor cells. So what you can see on the top right-hand side, we can mask over those areas and we shine UV light over that. We collect the barcodes, we do whole transcriptome, and you can collect protein information out of that as well. And you can flip that mask over to outline other regions of the tissue as well. So you can perform this interesting segmentation kind of analysis. So the tissue or those micromirrors are very robust. So depending on the structure or the area in the tissue that you want to profile, we have different modalities in order to accomplish that. So geometric profiling, we can segment between different compartments, as I mentioned. We can look at contour profiling to lay different tracks across the tissue. Or if your tissue is not contiguous, that's completely fine. If there's a diffuse set of cells, we can actually mask over those as well, as long as they're captured within that region that you define. So the types of applications for Geomix, you can refer to our website. We have over 200 publications on Geomix now. But I was just thinking about one of the original papers that we did. This is the first Geomix paper that we published in Nature Medicine, and this is coming out of Jen Wargo's lab in the U.S. and also James Allison, and he's a Nobel laureate, as many of you know. And what they were able to find using our platform was the tertiary lymphoid structures and the importance of response of patients to immunotherapy to tertiary lymphoid structures. So before they had used our technology, TLSs were not even defined. That's pretty exciting. So if we fast forward a year later, or 15 months later, after that, Jen Wargo and also another lab out of Lund University in EMEA, they discovered the same exact thing. So the importance of B cells and the composition of that tertiary lymphoid structure. And what we're finding nowadays, which is really interesting, it's not only the presence of tertiary lymphoid structures and the response of that patient to IO, but the actual structure and the composition of the cells within that TLS that's important for outcome. So this field was actually launched by our technology and enabled by Geomix. So as I mentioned before, we have whole transcriptome analysis for RNA. We also have targeted panels for cancer and others. On the protein side of things, we have over 300 antibodies that are validated for immunology and neuroscience. These are built out in modular fashion. And of course, if we don't have any content included in our validated and optimized panels, you can customize. So you can spike into our existing panels and or create standalone panels. So up to 400 plex for RNA. We've also recently launched our proteogenomics workflow. So now you can actually look at whole transcriptome profiling and up to 150 proteins on the same tissue section. We are the only technology that can get up to that level of plex for both of those analytes. You can scan the QR code here to have a look at the white paper, or please contact us. So at NanoString, we also have partnerships with other industry leaders. So in terms of sample prep and automation, we've partnered with Leica. So on the Bond RX, you can do your upstream tissue staining, which reduces your hands-on time to less than 30 minutes to get your samples onto the Geomix instrument. And we've also partnered with Illumina as well. And we use their Dragon BioIT pipeline to convert those NGS libraries into a file type that's recognized by the Geomix instrument and being able to use for analysis. So what that enables is 4 times more whole transcriptome analysis and throughput than any other competitor on the market. In H2 of this year, we'll be launching a new, faster and more diverse functionality on the Geomix instrument. So we'll be looking at using user-guided AI and driven ROI selection to automate that process. Some push-button solutions for reagent changes and to streamline the workflow efficiency. At AACR in April, next month, we'll be talking about the increase in protein plex. I highly suggest that you tune into this one. I've seen what it is, and it's absolutely going to blow your mind. Proteogenomics, as I just mentioned on the previous slide. And we'll also be launching our TCR diversity panel. So interestingly enough, a very high level and respected group leader and scientist in Korea found a very interesting discovery using this panel on our alpha program. Just last week, he discussed that with us. It's very exciting. So now I'll shift over to the CosMx SMI. So this is the new kid on the block, in which everyone is talking about. We're very excited about this. It's the most widely adopted single cell spatial imager on the market. As of the end of Q4 of last year, we had over 180 pre-bookings for this system. That has increased now that we're we are into March this year. Our partners at Philly Korea have booked some of these instruments in advance, so they will be available to the Korean market in 2023, should you want them. And this platform, even though we've just launched this at the end of last quarter, we've delivered over 110 customer projects. So those are customer samples that we run through our technology access program in Seattle. We've profiled over 70 plus tissues using our platform and profiled over 50 million single cells. And at the moment, we have 15 plus publications. I think 7 of these are been published, and we've got a few other in the pipeline that are under review. And these are coming out. And these are publications that are done on our commercial platform, not on any sort of pre-chemistry or anything that was developed in an academic lab before it was acquired. This is true discovery on the instrument that you'll be using. So if we look at this example here, this is an FFPE melanoma tissue that was run on a 1,000-plex RNA panel. You're looking at 4 million cells here and 2 billion transcripts in a single run. So for those of you who work in single-cell RNA-seq, consider this. It's 15% of the total number of single cells in the entire Human Cell Atlas project across 406 labs that were run in a single run, right? So that you can profile nearly 1 million cells on a single slide. It's absolutely incredible. And it can be done in a single week. So if you compare that to looking at running through single-cell RNA-seq, the amount of money you spend, the amount of time spent in that to get data that doesn't give you spatial information, we can actually do that for you with the level of increase in Plex as well that we're doing and enable discovery in a spatial context much quicker and much cheaper. So some key metrics for the CosMx SMI. We have the highest Plex. We have demonstrated over 1,000 Plex, and that's increasing, which I've already alluded to. And what I mean by demonstrated is that we have an on-market product, and we've delivered over 110 projects using that 1,000-plex panel. It's not something that we have in our roadmap. It's not something that we're aspiring to. We're actually developing and putting out customer outputs and data on that 1,000-plex panel. Like the Geomix system, you can process any sample, FFPE, fresh frozen, TMA, and organoids. We are high-plex multi-omic, which I'll discuss here, so we can look at RNA and protein, and even on the same section. And we have a tunable throughput. So depending on the assay that you choose, the area that you choose to scan, we can process anywhere from 2 to 20 slides per week. And we have a high degree of resolution, so 50 nanometers in the X and Y plane. So how does the CosMx work? So what we do is we take that 1,000-plex panel. We also have a 100-plex panel, by the way. I should mention that. But in all of those 110 projects that we've delivered, not a single customer has opted for a 100-plex panel. Everyone always wants the highest plex. And I know in Korea, based on our experience with Geomix, once we went from our 1,800-plex panel to whole transcriptome, every Korean customer wants whole transcriptome. And I know that's going to be the case with SMI as well. But anyway, so we take that transcript data, that 1,000-plex transcript data, and we also stain the tissue with protein markers. So these are not morphology markers per se, but we use those to define the cellular boundary, a nuclear marker, and we have some, pan-cytokeratin, CD45, and we have an a la carte channel open as well. But what we use that protein information for is to assist with cell segmentation. So we put that information into a cell segmentation. This is open source software. It's called CellPose, which some of you may be familiar with. And then we get accurate cell segmentation, and then we take that data. You can do 3-dimensional rendering with it, should you choose to, but typically we compress that data and we run a variety of different applications on that, which I'll show you in just a minute. So in terms of talking about cell segmentation, this is extremely important, particularly within a single cell in situ space. So you want to have the confidence that the transcripts and the proteins that you're calling into a particular cell are in the right place. So if you're using a nuclear inferred expansion model on your own, it's not giving you the appropriate boundaries that you require. And we are the only in situ company that's actually using those protein markers, as I mentioned. So we are not only using nuclei stain, cytoplasmic stains, and cellular boundary stains to inform where that cell sits in a three-dimensional structure to allocate those transcripts appropriately. It's extremely important, not only to do your cell typing, where those transcripts are located, but to do second order kind of analysis like differential gene expression and receptor ligand interactions. So we talk about Plex a lot here at NanoString, and the reason why we do that is what we found is that 1,000 Plex is the minimal amount of Plex required to do really comprehensive single cell spatial analysis. And that's why we started at 1,000 Plex. So if you consider low Plex type of applications where you're only looking at a couple hundred genes, you're really limited to the information that you can acquire. So typically the other products that you might see on the market, they'll give you cell typing information. That's about it. But what you need are more genes in order to understand these different functions within a variety of different tissues. And that's what we can enable. So in terms of application, what that 1,000 Plex panel enables is the ability to do appropriate cell atlasing. So we leverage a whole 1,000 Plex genes to do our cell atlasing, not just relying on a number of or a singular, 3 or 4 conical marker genes. You can perform neighborhood analysis using that. We can look at cell state and function, receptor ligand interactions, and also pathway analysis and biomarker discovery. And this is extremely hard to do, and it's very important as well that you have defined and accurate cell segmentation so you can look at differential gene expression within a three-dimensional plane. As I mentioned, we're also multi-omic. This is kind of a nebulous term in this space, but we have the highest Plex RNA, so 1,000 Plex panel where you can localize those transcripts down to a nuclear, cytoplasmic, or cellular boundary. And we also have one of the highest Plex protein panels on the market. So 64 Plex with the ability to do 8 custom targets and 4 morphology markers for a Plex of 76. This is a publicly available data set that we have available on our website to download. This was run on one of our alpha instruments, actually. But in this, we looked at the ability of our platform to work on real-world FFPE samples. So what are those samples that you're getting that are coming out of the clinic? So typically, we don't look at RIN scores or DV200 scores. We don't find that they are correlated with success on our instrument. But in this particular experiment, we had a look at them. You had DV200 scores of 60% down to 20%, which would not be compatible with any other in situ platform. And the RIN values were 2.4 down to unmeasurable. Now, despite those limitations and the poorness of quality of that tissue, we are able to have 94 to 97% of those transcripts past QC on our system. And again, you can have a look at this on our website. So there's been a number of labs that have actually taken that data set and they've turned that into publications. The first of which was Aviv Gev out of the Broad Institute in the US. And there's a variety of others that are developing new spatial methods for taking that, taking our data and doing some amazing things with it. So we're already enabling discovery with that open source data set. And at NanoString, that's what we want to do. We put all of our information out into the public domain, right? You're not guessing, we're not using a black box. The latest data set that we've produced is a human liver FFPE tissue. So we're looking at a normal and HCC tissue. So this is pretty exciting. And we're enabling more biological insight with this panel. So this is run on our commercial instrument. We're detecting over 1,150 mean transcripts per cell. To give you an idea, that's about 75% to 50% more than anyone else is doing currently. Within that 1,000 Plex panel, 70% of the targeted genes are detected across that tissue, or 700 unique genes, so a large degree of diversity across that. And we have a high degree of specificity, so 99% of those genes were actually on target. And we use a series of negative control probes and some false codes to make sure that we get accurate cell calls. And of course, I've already discussed at length about our cell segmentation, so you can have a look at that using the QR code. So when we compare these data sets to our competitors, so this is from publicly available information. When we look at sensitivity, the CosMx is enabling 3 times higher discovery in terms of, or 3 times higher for transcripts per cell. And the diversity of cells, or the unique number of genes above the limit of detection, is more than 2 times higher. And the rate of identified counts that are on target, or the specificity of our system, is the same as our competitors. So again, looking at the protein content that we have on the system. So at NanoString, we grew up in oncology. We were initially an oncology company. This is the panel that we developed first. So we know how to build these panels. We know how to optimize them. We have a high degree of throughput with this. So 1 slide per day at 100 centimeters squared area to analyze. As I mentioned, it will be customizable. So in H2 of this year, we'll be able to add 8 additional custom proteins to that particular panel if the panel doesn't have your targets. And we also have an expanding menu coming. So a mouse neuroscience panel and others will be launching sequentially throughout the year. So the CosMx system, we have a very large imageable area. So on a standard positively charged slide, you can profile 20 millimeters by 15 millimeters. Okay, and you can do a whole tissue scan. You're not limited to fields of view, right, or just a handful of them. You can scan the entire tissue as long as it sits within this frame, right, up to 300 millimeters squared. And again, we have 4 slides that will be sitting on our system. So 12 centimeters of scannable area in terms of the amount of tissue that you can put on. And as I already mentioned, a high speed for the protein, we can do 1 slide per day. So we put a lot of effort into validating these protein panels. This is not just done ad hoc. So we perform an initial screen here doing a single plex unconjugated screen with IHC. We have a pathologist review that tissue, and then we conjugate one of our barcodes to it, and then we have a pathologist review it again. And we look at the performance of those proteins in space. And this is no small feat to get up to 64 plex doing that. And we take all that. We take on all that effort for you, and we run that in our lab. So we have a high degree of sensitivity and specificity with those, and there's a high concordance between slide to slide variability, instrument to instrument variability, and also when we're incorporating our automated slide prep using the LikeaBond RX as well. In keeping with that protein story, so this is a colorectal cancer sample that we have profiled in our lab using that 64 plex protein. And what protein enables us to do is add more information that you can't really see with RNA. So we have full endpoint functional validation, that you can do to, validate what discoveries you're finding with RNA. You can look at post-translational and functional modifications and subcellular translocation or activation of specific events that are happening within the cell that are invisible to RNA. So if we consider this example here, this is from a clinical trial. This is a breast cancer sample that's taken from Gordon Mills' lab. So we work very closely with him. He helped develop the Geomix technology with Joe. And here we're looking at a cord needle biopsy. And in this little vignette, you can see it stained just with a number of particular proteins here that we're having a look at. But what we're really interested in is the translocation and phosphorylation of ERK, right? So on the right-hand side here, we can see the phosphorylation in purple here that you can see that's the phosphorylated ERK that's accumulating in the nucleus of these patients that are having breast cancer. So this is a real breast cancer patient. And then this patient was actually given a phosphorylation inhibitor. So in the follow-up that they did, they grabbed another biopsy from this patient following post-end treatment, and you can see a decrease in MAPK and ERK signaling following therapy, right? So effective use of that particular therapy for that patient, which is really exciting. So just talking a little bit about roadmaps. So, at AGBT, we've had a presence there since 2019, launching the Geomix system. And when we first started there, the Geomix, we were doing, I think, 20 times more than anyone else was. And that was at 84 plex genes in 2019 on the Geomix system. A year later, we scaled up to 1,800 genes on Geomix. And a year after that, we were at whole transcriptome. Last year, this is actually 6 months ago at AGBT, we talked about our 1,000 plex panel, which we've been running for, nearly 2 years now. Fast forward to this year, and just about 3 weeks ago or 4 weeks ago, we launched showed our 6,000 plex CosMx panel. And we're already running customer samples on that, and I'll give you a little vignette to what we're doing there. And next year, we're thinking about going up to whole transcriptome, or actually not just thinking about it. Joe Beecham truly believes that we're going to reach that, and Joe has never failed to deliver. So keep your eyes open for what we're going to be doing next year. So what this is enabling, the 6K panel, which will be launching in Q1 of next year, this will be a commercial product. It gives you a 6-time increase in RNA plex of what we're doing right now. It's more than 12 times more than any competitor is doing. We'll also be increasing our protein plex to 120 proteins, which we've also demonstrated as well, which will be available in our technology access program in Q4 of this year. So make sure you get in line for that. Have a chat with your representative from Philly, Korea. We'll also be launching more content. So looking at a 1,000 plex mouse universal RNA panel, a 1,000 plex human neuro, 64 plex mouse neuro, and also the targeted add-ons that I mentioned before. In terms of throughput on the system, so at the moment with our 1000-plex panel, you can process anywhere from 2 to 8 slides per week, depending on the scannable area and size of your tissues. Our 6000-plex panel, what we found is it takes 1.7 times longer a turnaround time than the 1000-plex panel, with a throughput of roughly 1 to 5 slides per week. And that's what we'll be focused on. And there will be improvements in that over time as we upgrade our chemistry. So when we consider the Plex Game, this is, this plot here. If you're looking at, sort of a low Plex kind of offering, all of the spaces that are colored in green is the area of biology that's covered with like a couple hundred Plex, right? So if we compare that to our 1000-plex panel, you can see the amount of biology that's covered just with 1000 Plex. So we've been using this already. We're looking at over 68 different tissue types or 70, 110 projects delivered, those 50 million cells and those 15 preprints and publications, all that are enabled on the 1000-plex. Moving into the 6000-plex, you can see the massive change in the amount of coverage that we get across the biology. And really, we're showing that there's no limit to what we can do. And this Plex is going to increase. So this is enabling more biological insight than any other platform, in situ platform on the market currently. So you might be asking, does this actually work on reference materials? And it absolutely is. So Joe Beecham invented a new NGS-based validation method that scales. And he's borrowed from RNA-seq here. And he's run this method looking at FPKM values over 35 different cell lines. And he finds that it scales extremely well. And our sensitivity is 1 transcript per cell and the specificity of over 98%. When we look at the performance of our 6000-plex panel to our 1000-plex panel, this is a run on some PDAC samples from a customer. This is the 1000-plex panel within, nested within the 6000-plex panel, and you can see that there's a high degree of concordance between the two. So they are scaling, and we're having no issue with that whatsoever. So it's completely possible, and we are using it. So I'll give you a little vignette into, can this 6000-plex panel actually work on customer samples and real-world samples? The answer to that, of course, is also absolutely. And this is some extended work that we did for Will Huang. So he's out of the Broad Institute and Mass General. And this is building on a study that he had using the Geomix that reached the cover of Nature Genetics late last year. So here's some raw images of that panel. On the left-hand side here, we have an 8-plex protein image within that sample. And in the middle there, this is the raw RNA image, a transcript image that we have. If we blow up that sample right there, you can see on the right-hand side all of the different cells that are segmented, and you can see that they're just absolutely jam-packed with RNA. And from what Joe told me and has told us, that actually when he built this 6000 panel, it didn't take much for him to actually decode that. He said he just kind of built it and it worked because of all the effort that we put up front on that 1000-plex panel. So looking here, this is an 8-plex protein image that was done on the same exact slide. So you can see the nice, beautiful architecture of the tissue, which, protein does extremely well. But we started to look at this and, basically Joe was saying, we got to make sure that we can actually detect these transcripts. Otherwise, if it's just a blurred mess and it's not working, then there's no point in building it. But fortunately, what he found is that it does work and extremely well. So what he was finding is that when, the counts, once you get up to about 50,000 cells, you find that about 4,800 to 5,300 unique genes are actually detected within each of those cells, 3 logs over our negative control probes. So we use ERCC negative control probe sequences. We do this with Geomix and also false codes. And what we're finding is a high degree of detection here. So if we did dive into that particular data set, what are we actually finding? So, on the left, of course, you get your cell type distribution. And on the right-hand side, you get your UMAP, right? So this is where single cell ends. This is where we just begin. So you can see a nice distribution of all of these individual cell types. So this is across 5 different patients. And we're looking at different tumor types as well. So, with our platform in that 6000-plex, we're able to identify unique patient tumors within that area. So this is absolutely incredible. And this is something what we're doing. So if we do a little bit of a deep dive into what's possible with that 6000-plex, I'll show you here. So we're really opening up a new realm of spatial discovery that is unprecedented, right? Nothing like this has ever been seen before. So if we consider signal transduction, just for an example, right? This is one bin of biology that we can look at. In step 1, you pick any class of molecules you want to spatially explore. So we're picking signal transduction and specifically signaling ligands. So in our 6,000 plex panel, there's 407 signaling ligands that we have. And we spatially correlated those with the 23 samples from those PDAC samples. And we're looking at the interaction of those cells in space. So we can do that very simply using our AtoMx pipeline as well. So we have just drag and drop modules that we can use through our pipeline orchestrator to enable the discovery on the platform. And this is something interesting that we found. So this was developed by an algorithm that was developed by Patrick Donaher out of our lab. And we're looking at the correlation of specific ligands in the tissue. So in the middle here with this correlation matrix, we have the different ligands. And what you can see is across those, each individual sample in those ligands, you can see that there's hotspots of these co-expressed ligands within the tissue. So what we can do is we can actually blow up where those particular clusters are showing up within the tissue. And what we found is some really interesting biology looking at chemoattractants and chemokines within these individual hotspots. And as far as we know, this is the first time this has ever been done in space. So looking at more of a deep dive into actually what that means, we're looking at these individual hotspots within the tissue. We can actually identify what particular cells are talking to each other. So we're looking at those receptor ligand interactions, looking at cancer-associated fibroblasts and macrophages. They talk to each other in about a 70 micron radius. If we move over and look at cancer-associated fibroblasts, macrophages, and malignant cells, that's a tighter clustering of about 40 microns. And again, looking at macrophages, nerve cells, malignant cells, etc., any single hotspot you can look at, we can identify how those cells are arranged in space and how they're talking to one another. Looking at these samples as well and looking at these individual clusters, what we can also see is that these types of communications and interactions that are happening across the tissue are quite diverse. They happen in all of those hotspots, but of course, the composition of those cells within each of those hotspots completely changes, right? And how those patients are behaving and their responses to therapy or progression of disease changes based on what we're seeing in the tissue. And again, this is only scratching the surface. You can go back to all of that biology again, and you can enable all of these different kinds of discoveries with this level of Plex. This is the last slide I'll show here for the CosMx. As I mentioned, we'll be getting up to 120 Plex, but this is an experiment that we ran across a variety of different tissue types. And you can see here that we're getting up to 120 Plex, and we'll be able to answer some really interesting information as we start to really dive into the biology and figure out actually what's going on here. But we're certainly showing it here. The last part of my talk here, I'm just going to talk about our AtoMx platform. So this is our spatial informatics platform. And I want to reiterate that this isn't just the software. It's actually an environment and a platform that provides you with a lot of flexibility to not only visualize your data, but to store it and do a variety of different things. So you can do, as I mentioned, all of your data analysis and visualization can be enabled through the data analysis pipeline that's hosted on AtoMx. All of these data sets can be exported as a Seurat object, so you don't have to do that and turn these complicated data sets into that. You have scalable compute and data storage and management. So this is done, the cost overall of using the AtoMx platform is roughly 10% of your overall reagent spent, right? And it's not going to be an onerous type of platform. So we're not interested in making this, not making it cost effective, essentially. You can do easy sharing and collaboration as well. So if you're working with different groups across Korea or collaborators in APAC, EMEA, or the US, what you can simply do is send that individual a link. They can follow that link via email, and they can be embedded in these massive data sets. So if you're looking at all of that imaging information, which of course is really important for using third-party type of tools to run through an analysis pipeline, it's very difficult to share that data. So if any of you are doing that right now and sharing imaging data, you're using AWS infrastructure, which is exactly what we're using here. There's a high level of security and compliance associated with this. So we have major banks and pharma are using the same type of platform, and it's also cost effective as well. So I've talked about that. When we compare this to our competitors, right? So not all softwares, quote unquote, are actually built the same. So not only does our platform enable an interactive imaging and data viewer, but it also allows customization and data analysis pipelines. So if you actually consider how this would be deployed in your laboratory, you have different functional groups within your lab, right? So you have your wet lab scientists, you have your postdocs, your PhDs, you have your data scientists and your bioinformaticians. So your bioinformaticians certainly will use, Python, R, whatever it is, to develop their own custom modules, and they'll be doing that in the background. But they can develop those pipelines, and then they can actually import them as modules into the AtoMx platform. So that way, when they build these pipelines, other members of your lab can simply build the pipeline, press go, and you can see in real time how that is being deployed, and that frees up your bioinformatician and their analysis pipelines to do other work. It's inexpensive. As I mentioned, it's only 10% of your overall reagent spend, and you have on-demand compute power to scale according to your needs. It's a hub for multi-site collaboration, as I mentioned before, and you have long-term glacial storage that you can access anywhere at any time. When we look at the pipeline orchestrator, so this is something that I mentioned before, these are just drag and drop analysis pipelines that you can use, and you build these completely out. You can share these with your collaborators. What's also interesting as well is that as you start these modules, you will get an on-instrument prompt to tell you how long it's actually going to take to run the analysis. So it's my understanding for bioinformaticians, when they start these pipelines, they don't exactly know when it'll be finished. It could be 5 hours, 18 hours, whatever it may be. But with our platform, you can see that in real time. And I know that we're improving the data analysis time, going from days to simply hours. So you get a high degree of resolution. You can do accurate cell segmentation and have a look at that and be able to run that through our pipelines as well. Within the pipeline orchestrator as well, as you run through certain modules, you can have a graphical readout of what that looks like in real time on the right. So here's an example of a UMAP clustering that you're familiar with. In this particular module here, we're looking at protein expression and how that's panning out within a particular area of the tissue. And you can scan over that in real time and see different areas of the tissue and how the analysis is going. As I mentioned as well, it's completely customizable. So your bioinformaticians can upload their own scripts, or you can download publicly available scripts from Bioconductor, and you can integrate those into your pipeline. So throughout 2022, what we'll be looking at is publishing our source code. So we're not going to hide what we're doing. We'll provide you with everything that we're doing, and we'll make it publicly available so you can use it. And we'll be expanding our CosMx data analysis, through methods that we develop, and also through methods with our partners. And hopefully we can work with some of you in Korea. I certainly look forward to that. In H2, for those of you who are not interested in using a cloud solution and you believe you have the infrastructure in place, that's fine. We'll have an off-cloud CosMx solution for you in H2 of this year. And rolling out later in the year as well, we'll be able to integrate Geomix data analysis into the AtoMx workspace. With Geomix as well, we'll also be looking at integrating third-party analysis tools as well, and incorporating more of the pipelines that we see from the academic community. So that's everything that I have today. I hope that you enjoyed the updates that we're providing. I know at NanoString we're extremely excited about 2023 and what we're ushering in. If you are interested in talking further, please reach out to me or one of your representatives from Korea. And I certainly hope that I get to see some of you next month. So thank you so much for your time. I really appreciate it.